last three chapters, we've talked about the metabolism of acetyl-CoA through the citric acid cycle and the electron transport chain, how carbohydrates can be used for energy aerobically with our central metabolism or anaerobically without it, and how carbohydrates can be stored for later. We've also talked about how fatty acids can be used or stored and how they supply energy differently than carbohydrates. In this chapter, we'll talk about how amino acids from the dietary intake of protein can be used for energy and how they are very different than fats and carbohydrates in their use. Again, we want to think about energy from proteins as they enter our body, as proteins in foods. Proteins will be degraded to amino acids before they enter the bloodstream, and the amino acids will need to have the nitrogen removed before they can join in with the other pathways. Proteins, like triacylglycerols, are not particularly accessible to enzymes. As the protein is typically folded with tertiary structures when we start to eat it, it's difficult to get any access for the enzymes until the proteins denature in the stomach. Once denatured, it's important to start to cut up the protein with proteases, so the protein can't renature in the intestines. In the intestines, many proteases are present to cleave the wide variety of amino acid bonds available. Proteins are hydrolyzed all the way down to individual amino acids, which are then taken into the intestinal cells and then passed to the bloodstream. Again, the important steps in digestion all happen in the stomach and the intestines. The acidity of the stomach allows proteins to be denatured or unfolded, and pepsin, a specific protease in the stomach, will break apart the primary structure so they won't refold. In the intestines, many more enzymes will break the peptides all the way down to individual amino acids, which can be transported through intestinal cells and into the bloodstream. So when we look at protein supply of energy, it actually enters into our bloodstream as amino acids, and that's what we'll use for all of our following processes. Once amino acids are in the bloodstream, there are a wide variety of possibilities for how they may be used. However, with amino acids, there's no way to store the amino acids for later. So we have a relatively small pool of amino acids that are held in the bloodstream in the cells. This is tightly regulated, so amino acids ideally are always available for the type of work for which we need them. But that also means whatever sources that we have for the amino acids need to be balanced with some use for the amino acid. Sources like diet, protein turnover, or synthesis, which we can see on the left, are always balanced with uses like making proteins or making different amino acids or nucleic acids or being used for energy and waste, the types of things we see on the right. Since amino acids are really the only way that we can get nitrogen, we call all this balance of amino acid sources and uses the nitrogen balance. We talked about essential amino acids some in chapter 20 when we started talking about proteins, but in this chapter we'll talk a little bit more about why some amino acids are non-essential and some are essential. The main idea here is that many of our amino acids are really closely related to either other amino acids that can be used to synthesize them, or closely related to central metabolites that can be converted into amino acids, for example, pyruvate, which can be made to alanine. We accomplish this conversion of central metabolites by a process called transamination. That really just means transferring an amine from one molecule to another molecule. Our bodies will transaminate for two different reasons. Either we need the nitrogen someplace else, so our bodies can use it to either make non-essential amino acids or neurotransmitters or nucleic acids or whatever else that has nitrogen in it, or we're going to use the carbon portion for energy and we need to get rid of the nitrogen. Either way is a reason for our bodies to transaminate. Your book talks about these separately, but it really is the same process for each. Transaminations always work by swapping a ketone on an alpha keto acid with an amine on an alpha amino acid. Remember that the alpha always refers to the position 1 over from a carboxylic acid or a carboxylate. So an alpha keto acid has a ketone 1 carbon over from a carboxylic acid or a carboxylate, and an alpha amino acid has an amine group, a nitrogen, 1 carbon over from a carboxylic acid or a carboxylate. Make sure that you can identify both a ketone, which you can find in chapter 15 notes, and an amine, which you can find in chapter 17 notes. But also make sure that you don't get a ketone mixed up with the other carbonyl-containing compounds, specifically carboxylic acids and carboxylates, because those are always also going to be present in these compounds. In every one of these cases, the only thing that happens in a transamination reaction is that the amine and ketone are going to be swapped between molecules. This can be really helpful since many of our central metabolites are alpha keto acids and can be easily converted to amino acids by transamination. On the right, you can see the relationship between pyruvate, the product of glycolysis, and alanine, an amino acid, or oxaloacetate and alpha-ketoglutarate from the citric acid cycle, and the amino acids aspartate and glutamate as well. When we are performing transaminations to separate the amine and the carbon portion for energy, almost all the transaminations will be specific for the keto acid, alpha-ketoglutarate. That's important because that means the product is always glutamate, and the glutamate can be transferred to the liver as the entry point into the urea cycle, which allows us to store and ultimately eliminate excess nitrogen with urea. Our muscles tend to transaminate pyruvate to alanine instead. So instead of moving the nitrogen onto an alpha-ketoglutarate and making glutamate, we move them onto a pyruvate and make alanine. And that's largely because this can both transfer a nitrogen, but also facilitate gluconeogenesis in the liver at the same time. Remember that gluconeogenesis primarily occurs in the liver and uses either lactate or pyruvate as the source for the new glucose. In the liver, the alanine is converted back to pyruvate, and then that can be used for gluconeogenesis, and then ultimately glutamate is formed with the nitrogen. Once glutamate is in the liver, it can be converted back to alpha-ketoglutarate by oxidative deamination. This process of oxidative deamination frees up the alpha-ketoglutarate to receive more amines back in other tissues and also produces ammonium. Ammonium is actually relatively toxic, so our bodies will need to deal with it fairly quickly. This process is also oxidative, and the reduced cofactor produces stored energy. We can either use NAD+ or NADP+ as our oxidizers. When we use NAD+ as our oxidizer, the NADH that is formed can power the electron transport chain. We can get some energy out of it. When NADP+ is used, the NADPH that is formed can power lipogenesis. Serine and threonine can be transported to the liver directly and deaminated through a non-oxidative pathway. This still gives us ammonium, so the big takeaway is that even if we deaminate through a different route, we still have ammonium that we have to deal with. So whether we're getting ammonium from glutamate, serine, or threonine, we ultimately can't hang on to it. Ammonia and ammonium, conjugate base and conjugate acid, are both toxic. Fish just pee it out, but that gets awkward for humans. So we have the urea cycle, which converts the ammonia to a less toxic form, urea, which you can see on the top right of the slide. Urea is actually the amide version of carbon dioxide, ultimately coupling two toxic ammonias with a waste carbon dioxide and removing them all together. The urea cycle starts by coupling the first ammonia onto carbon dioxide and then activating it with a phosphate. The resulting carbamoyl phosphate is actually what joins into the urea cycle, and it costs 2 ATP to make. Several amino acids will be used in the urea cycle, including arginine, one of the 20 amino acids that are found commonly in human proteins. But we'll also use ornithine and citrulline, which are amino acids that can't be encoded genetically. Surprisingly, the citrulline, which you can see on the bottom, it looks mostly like urea on the left side, is the least related chemically because the nitrogen-carbon bond there can't be easily broken. The carbamoyl phosphate couples its carbamide, that's a carbon double bound to oxygen plus a nitrogen bound to that same carbon. It couples that carbamide group onto ornithine instead of the mitochondria, resulting in a citrulline that can be transported outside of the mitochondria. Now the citrulline looks like it has a urea on it, but we can't break off that urea very easily. In order for us to actually eliminate the urea, it needs another nitrogen, which we get from aspartate. This costs an ATP being converted to an AMP, so an adenosine triphosphate going all the way to a monophosphate, which is the equivalent of losing 2 ATP. The aspartate brings along its carbons to do this though, and can be eliminated as a fumarate, which is an intermediate in the citric acid cycle. At this point, the resulting arginine can be converted to urea directly, and ornithine, which can go back into the mitochondria to be continued on as part of the urea cycle. The urea can be stored until later when we pee, and the ornithine continues to be cycled as it joins on another carbamoyl phosphate. If the structures are more your style, this page shows the transitions. A carbamoyl phosphate installs the carbamoyl group, again a carbonyl and amide, into an ornithine. The carbonyl gets converted to a nitrogen by attaching aspartate at the cost of an ATP being converted to AMP, but then the aspartate gets removed as fumarate and leaves behind the nitrogen. The group can be hydrolyzed off and the cycle repeated. Notice the net reaction on the bottom of this slide. 1 ammonia and a carbon dioxide get processed with 3 ATP to make urea and 2 ADP and an adenosine monophosphate, an AMP. Because we have one transition from ATP all the way to AMP, the net reaction is actually worth the same as 4 ATP to ADP, adenosine triphosphate to adenosine diphosphate transitions. If we look only at the urea cycle, it looks like we spend 4 ATP to get rid of a single ammonia, which seems kind of expensive. Our saving grace is that we have to also convert aspartate to fumarate, which we can actually get some energy out of. It doesn't actually cost us as much as it looks like because the urea cycle and the citric acid cycle tend to operate in tandem. The fumarate that's formed in the urea cycle can be hydrated to malate and oxidized to oxaloacetate, some of the last steps of the citric acid cycle, which gains us an NADH, or the equivalent of 2.5 ATP. That oxaloacetate can then accept an amine via transamination to become the aspartate that we can use in the urea cycle. For this reason, we're actually processing 2 ammonia, one from the carbamoyl phosphate and the other directly from glutamate via the aspartate. We're also gaining the equivalent of 2.5 ATP from the citric acid cycle and the electron transport chain, at the same time that we lose the equivalent of 4 ATP in the urea cycle. So the true cost is actually 1.5 ATP for every 2 amines that get processed, or a little less than an ATP per amine.